Good morning. Thank you for signing up with us here in SunUP on 7. Wherever you are, thank you so much. If you're just tuning in, I want to resonate that welcome. Earlier, we were having two different conversations. We were talking about child sexual abuse and the services offered by the Ministry of Human Development and Child Protection Services. Then we jumped into the conversation with the Labor Department and Ms. Amy Jex, talking about the mental health forum that's happening tomorrow, which is a very important conversation because they're actually pushing for the conversation on mental health and mental well-being, especially for frontline workers. If you want to follow that conversation and tune into the forum is happening tomorrow starting at 9 in the morning and it finishes at 3:30, and they're going to be talking the different aspects that can affect the mental well-being of frontline workers. So follow their Facebook page and then you can find the link to their Zoom forum. So participate in that conversation. It's so meaningful and it's so holistic. And continuing with our health conversation, we're joined by a viral immunologist who has graced us on his vacation in Belize to be here with us on SunUP on 7 to talk about viruses. And in particular, we're going to draw it to COVID-19, how the viruses work, how mutations work, and how these different mutations can impact our immune system and our well-being. So, Mr. Chinedu Ugwu, did I pronounce it right?
Yes, I think.
Okay, no, if not, you can correct me. Please do.
No, I think it's the accent that is different. Is Ugwu Chinedu.
Ugwu Chinedu?
Yes.
There you go.
See, okay.
Yes. So he's a PhD student, and you study at Oxford and Cambridge University. And like I said, you're here on vacation.
I know, I love guys, on the commercial break, I just have to pinpoint that Mr. Chinedu?
Chinedu.
Yeah, Chinedu.
Yeah, okay. So, Mr. Chinedu, we're going to get this by the ending of this episode, of this show, okay? Chinedu is a good husband because he's he's on vacation, but his wife said he should be on the show, so he's being on the show because of that. And that, all husbands out there, pay keen attention. Follow what your wives are saying. That is some good stuff right there. So we're thankful for your wife pushing you here, and we're thankful for your work too as well. We understand you're researching on COVID right now. Could you share some of the findings that you've had thus far?
Thank you. I've finished, so I have my PhD from.
Congratulations.
Oh, congratulations.
So what we're doing is, after my PhD, I was involved in a project that is funded by the UK British Research Foundation in England, BBSRC. Okay. So they require us to understand, there's a big viral problem we have in Nigeria called Lassa fever.
Okay.
So they require us to understand the diversity of the viruses that causes Lassa fever and how to understand the immune protection. What are the features of your immune component that protects you?
Okay.
So that project took me back to Nigeria. So having been on that project, then COVID happened.
Ah.
So when COVID happened, so we're also lucky to apply to Cambridge, Africa, and we got funded to do similar research on COVID. In this case, what we are to do is to look at if you survive COVID naturally. So if you got COVID and you survive, what features of your immune system help you to survive? So for example, we have what we call antibodies. Your antibody that help you to survive is your T cells. So that project is ongoing now. That's what I'm doing in Nigeria. So we recruit survivors of COVID and we look at their immune component. And the aim of the project is to, if we're able to understand features of natural protection, we then can predict what do you expect from when you get vaccinated. So if you are given vaccine, what kind of immune response you should make, and what is the standard immune response you should make that we can use to quantify the efficacy of vaccine. So if when you hear in the news, Pfizer is 95% protective, AstraZeneca vaccine is 74% protective. What we basically look at the immune response that vaccine can induce, and then we can quantify it. We can use numbers to say, okay, this can protect you 95%, this can protect you 90%. So what my research is doing is to understand those things we are looking for, so that when you bring Pfizer vaccine, we can then say, okay, that Pfizer vaccine, for us to know that Pfizer is effective, you should give us this level of antibody. It should give us this level of T cells. That's one of the research we are doing. On that research, we're also lucky to get another grant to look at, now that the variant, COVID variant became a thing and is fast spreading more than the actual COVID. So we got a grant from Gates Foundation as well. And what we are meant to do is, what is the implication of this variant on the immune response? So you have the Alpha variant, you have the Delta variant that has been a big problem, you have the Beta variant that came from the one from South Africa, then the Alpha variant from the UK. So the aim of the Gates Foundation research is for us to look at, so these different variants, how do they impact the immune response? For example, the ancestral COVID, the first COVID wave that came up. So let's say if that COVID is able to, the vaccine we make can protect you maybe 90%. So how does the Delta variant do? Does it also protect you? Will the vaccine be able to protect you against the Delta variant 90%? Or will it reduce the protection to 60%? So we have a system to study we call it a neutralisation assay. So what we do is we collect samples from survivors and those that we are vaccinated. And then, if you've been vaccinated, you should make immune response. So we assume that you have immune component in your sample, in your blood samples. Then we'll engage your immune component with what we call pseudotype virus. It's not actual virus, but we'll design something that looks like those variants. So we'll test your immune component. It's just like setting a wrestling match between your immune component and then all the different variants, and we'll see which one can win. So if your immune component is able to knock off all the different variants, then we are confident that the vaccine is able to take them all out. If maybe it can take out the alpha variant and can take out the beta variant, but may not do well with the delta variant, then we can inform those the vaccine producer that, oh, we need to tweak this thing, we can do it better. So those are the few researches I'm doing in Nigeria now.
So I wanna tap into some of your findings in your research. I love to hear. I always tell Renato I love viruses because they're so cool and how they mutate to survive. And I want to now tap into your findings. So you're saying that you've been working with people that are survivors of COVID. So let's talk about people that, for instance, I had COVID earlier in the late last year, and I also got vaccinated. So how does somebody that had COVID, what level of immunization they have or immunity, and how does the vaccine contribute to them being better prepared to handle COVID?
That's an interesting question. You see, you are one of the people who could want to look at your immune response as well. You know? So it's an interesting fight. So these are things we are learning. Because I think one thing I think people should give the scientists credit is that COVID is a novel thing, and we are trying to understand what is going on. So it's more like understanding and also trying to solve emergency problem. So with what you ask is, so what we've seen is, so I'm going to try to simplify how we make vaccine. So the way we make the COVID vaccine, if you imagine I'm the virus, I'm COVID. So all of the vaccine is targeting this shed. So COVID has, the same way you wear shed, it has an external envelope that covers the virus. It also has the internal protein that also help the virus to thrive. The same way you have your liver, you have your heart, all those kind of things. So most of the vaccines, almost all of them, are targeting your shed, which is the S protein of the virus. So if the idea is that if you can recognize, I'm different from the two of you because I'm wearing a white shirt, right? So you could recognize me by my white shirt. So the idea is that if I give the immune system this white shirt, they will recognize. If COVID is wearing a white shirt and I give it to the immune system, everything wearing white shirt will be recognized as a bad guy, which is COVID, you know. So the immune system will make antibody or T-cell respond to that white shirt.
Right.
So that's what the vaccine is. But that would be a different situation when you are infected. So when you are infected, you only don't have white shirt. You are infected by me. Everything about me, both my liver, my heart will infect you, right? So you're able to mount immune response, not just to the white shirt, but to my liver, to the heart. So what I'm trying to say is, so if the COVID virus, every vaccine is targeting the S protein, if you have been infected and you're a natural survivor, you will not just have antibody response to the S protein, which all the vaccines have. You also have antibody response to maybe to the nucleoprotein of the virus, like the same way the liver, because you were infected by the actual virus.
Right.
The vaccine doesn't contain the actual virus. The vaccine contains something you can recognize. The immune system can recognize.
Thanks for that clarification.
So the advantage you have now is that you're able to make immunity to both the nucleoprotein of the virus, to the S, which the vaccine also helps you to make. I don't think we know confidently whether it protects you better than just having just the vaccine. But your body is if you survive, your body it's like you have all of the ammunition to just fire.
Better prepared.
Something like that. But we don't have clear data that shows that because you were infected and you are better protected than the person that is vaccinated. We are confident that the vaccine will protect you, especially from hospitalization. But for example, part of what I'm hoping to find out from some of our research is to see if you're a natural survivor, maybe that protects you from reinfection compared to vaccination. Those are some of the things we hope to see from this data. Well, because we collect samples from natural survivors and also from vaccinated people. So in the long run, we want to compare how does your immune response as a natural survivor. So maybe we collected your sample today. After 6 months, will our natural survivors cohort and compared to the vaccinated cohort, which one got reinfected from the virus? Okay. We don't know, but these are some of the things we are hoping to find out along the line with the research.
This is some interesting research.
So, unfortunately, we're running out of time. If somebody wants to read your articles, your research, where can they find it?
You could go to the lab website. So I work for a research center in Nigeria and the university. It's called African Center of Excellence for Genomics of Infectious Diseases. Okay. So if you go there, you see most of the work I do. I'm also very active on Twitter. I don't know if you're able to pronounce my Twitter name, but it's called Ojemba Emulo 85. You will see my handle. But if you go, every work I do is always on the lab handle on social media. If you go to SGID website, you will see everything we do and all our research. And one more thing, if you also go, because my PhD was in the University of Cambridge, so we'll have a long-term collaboration with Cambridge and the work I do in Nigeria. So if you go to LVZ Lab, University of Cambridge, that's where you see most of the research. I'm involved in Cambridge as well. And the thing one thing I didn't say here is that in Cambridge, we're also involved in vaccine production for COVID. And the unique thing about the vaccine we make, which will be in clinical trial by next month, is the unique thing, what makes our own vaccine different is that we are using the sequences, the viral sequencing from outbreak. So what we do is, now all these variants are emerging. So we don't just use the old COVID virus that started in December 2020. We use the current sequences available in the outbreak. So our hope is that you are better off protected regardless of what comes up. Because we intend to use computation to look at all the variants, and then all the information we got from the invariant is imputed in the vaccine production as well.
I love to hear that. So it means that we'll have a better chance at fighting COVID and, reaching some kind of end to this pandemic. So I want to thank you so much for coming to share your research. Again, if you want to know more about the work that Chinedu has been doing, find out at the African Center of Excellence for Genomics of Infectious Disease. So with that, again, thank you so much for being here with us. We're going to go on a short commercial break, and when we come back, we're going to be talking about massages.